It's my pleasure to introduce our speaker for today, Kevin Janes, who is a professor at the University of Virginia and is a leading researcher that combines experimental and computational methods in not only cancer biology, but signal transduction in general. Kevin is a graduate of Johns Hopkins University, and after spending time in Spain as a Fulbright scholar, he went on to get his PhD at MIT, and then was a postdoc at Harvard, and before starting his lab at the University of Virginia. I connected with Kevin about a couple of years ago when he and his colleague Matt Lazzara were about to start a new multidisciplinary research center funded by the NCI at the University of Virginia. We discussed at that time the possibility of collaborating on some of the computational and modeling studies that he was planning to do. The center is called the Systems Analysis of Stress-Adapted Cancer Organelles. So the stress-adapted cancer organelles are a very interesting constructs that have properties that make them both difficult to analyze experimentally and model computationally. Various customized PD solvers are required to analyze some of the liquid-liquid phase separation that occurs in these organelles. So we started collaborating in more earnest last year when he actually also visited us. He came to the ICSB conference in Hartford last fall and had a couple of his colleagues join and visited us. And he is one of the sort of beta testers of the newly developed capabilities of the virtual cell to run as a containerized standalone simulation tools in command line form. I'm looking forward. I'm not sure exactly what he's planning to tell us from the many interesting things that he's doing, but I'm looking forward to his talk. Kevin.
Thanks very much, John, for the kind introduction, and thanks to you, Les, Michael, and Pedro for the invitation.
Is that just me? Sure. Is everybody?
We just lost Kevin.
Yeah, Kevin?
Yeah, we definitely just lost him.
Check that this is okay.
Oh, there you go.
There you are.
We lost you for a minute or two.
Oh, for a minute or two,
Well,
but
for
I'm
maybe
back.
30 seconds.
Okay, but everything looks good now.
Yeah, it's all good.
Yeah, it's all good. We just lost video and sound for 30 seconds.
Okay, great. Well, I'm here. Slides appear to be working. And as Jan said, my lab is in the University of Virginia, but I'm also here as a representative of the SASCO Center, which is a U54 research center sponsored by the NCI, focused at the systems level about how subcellular compartments adapt to the stresses related to oncogenesis and different cancer types. I'm not going to dwell on the center too much, but I'll simply refer you to our website here that describes many of the ongoing research activities, supplements, outreach type of things that we do. I'm here today to focus on one aspect of the overall center structure, which is this first research project on the regulation of chromosome segregation and the misregulation of chromosome segregation in cancer, which is a project that is co-led by myself and Todd Stukenberg at UVA and the Department of Biochemistry and Molecular Genetics. project focuses broadly on the regulation of proper chromosome segregation during the metaphase to anaphase transition during mitosis, as coordinated by a specialized protein complex in cells called the chromosome passenger complex. We've all taken even high school level biology. We'll know that as cells progress through mitosis, it goes from interphase, nuclear envelope breakdown, and prophase. During metaphase, the formation of the mitotic spindle that is important, crucial for ensuring proper segregation of the sister chromatids, and then progression into anaphase and telophase. The CPC plays a crucial role during metaphase in detecting whether or not duplicated sister chromatids are properly attached at the mitotic spindle, so-called amphitelic attachments, where there's one connection on either end of the spindle poles. When all of the mitotic chromosomes are properly aligned in this way, then cells can proceed into anaphase. The CPC accumulates in the inner centromere of metaphase chromosomes when that chromosome is not properly aligned and properly connected, and then has a low level of recruitment, or that recruitment disappears, and that is part of the signal for cells to progress into anaphase. When the CPC is recruited to the inner centromere, it sends additional signals that maintain chromosome cohesion, arrest cells at this phase in the cell cycle, enables cells to repair these attachments and try again to establish an amphitelic attachment, and also aids with chromosome alignment. And I already mentioned the progression through mitosis. To give a live cell example of this, this is a HeLa cell about to undergo mitosis. We'll see the chromosomes beginning to condense, nuclear envelope breakdown into prophase. And then as the cell goes into prometaphase and into metaphase, I'll draw your attention to 2 or so lagging chromosomes here that haven't properly aligned. And then what we'll see is that there's some delay. They will eventually get pulled, there we go, into the metaphase plate. And that very rapidly establishes the signal for the cell to progress into anaphase. There it is, telophase, and into cytokinesis. And so we'd like to try to understand at a molecular level and a mechanistic level how that sensing and activation of the CPC takes place using mathematical models that encode the reactions that underlie that network. What are some of the key features that regulate the recruitment of CPC to the inner centromere? There's 2 that are on the quantitative side of things, related to avidity and positive feedback. The CPC is a heterotetramer of 4 proteins. There's a protein kinase called Aurora B, which is important for regulating its activity, and also some of the other processes that I'll touch on lightly. There's a long linker called INSEMP, and then 2 subunits that directly or indirectly bind different histone modifications that occur on chromatin during metaphase. The first modification is set up by a kinase called haspin that's localized to the cohesin stripe that keeps sister chromatids together during metaphase. And then the second histone mark is established by a kinase called BUB1 that emanates from the kinetochores, which are the landing pads for where the mitotic spindle is trying to establish those amphitelic attachments. And the conceptual model that Todd has been working with for a number of years is that these 2 orthogonal marks, one of which occurs vertically and the other occurs horizontally between sister chromatids, create a bullseye in the inner centromere where the CPC can be recruited both by binding of both of the phosphohistone marks. Each one of those interactions is thought to be relatively weak, but in the region where they're both together, the CPC can bind 2 times, and this dramatically increases the accumulation, as illustrated in this immunofluorescence image of a metaphase chromosome. Now, the network overall that regulates CPC is beyond just those subunits that I touched on before. And what I'd like to begin with is how we ended up converging on VCell as the preferred modeling tool to be able to build this systems abstraction of the network. And it relates a lot to the notion of reaction and diffusive processes, particularly those that occur on chromatin. This is a paper not by us, but one that's out in the literature looking at the diffusion of chromatin using a GFP-fused TET repressor that's binding to randomly integrated TET operator elements in the genome of these cells. And by looking at the wiggling and diffusivity of the GFP, one can estimate effective diffusivities of chromatin-bound protein species. And what this paper showed is that whether the cells are in interphase or mitosis, the order of magnitude chromatin diffusivity is approximately 10 to the minus third micron squared per second. I'm in an engineering department, and we like to think about length and time scales a lot and trying to frame different modeling abstractions. And so given this effective diffusivity, we can consider the characteristic time for diffusion just in one dimensions for simplicity. Say the distance between those two kinetochores, that is roughly estimated to be about 700 nanometers. The length scale, or excuse me, the time scale of diffusivity to cross or to explore that 600 nanometers in one dimensions is simply 2 times the length squared, and it's 2 because it can wiggle in the left or it can wiggle in the right, divided by that diffusivity. And if one crunches the numbers, we get an effective time of about 6 minutes to span that 1D length scale. 6 minutes is important because the characteristic duration of metaphase is somewhere between 2 and 10 minutes, depending upon how long it takes to resolve those improper attachments. Therefore, we cannot ignore diffusion on chromatin because it's occurring at the same time scale as the process that we're seeking to study. And I'll contrast that with cytoplasmic diffusivities, which are normally on the order of about 10 microns squared per second. Here, this 10,000 times faster rate, we can effectively assume that any cytoplasmic species in our models are well mixed. Even though we do encode the diffusivities, we've never reassuringly seen anything remarkable there because that's happening much faster than the time frames that we're modeling. But we can't do that with the chromatin-bound states. In building this model of CPC regulation, we learned a number of interesting things, and I wanted to put these on the slides to give illustration of the value of model building in general. When Todd and I started working together, we framed this network as something that was relatively well understood, and we could try to recapitulate it with these modeling activities. But as we got into more of the details, we realized that many things that were thought to be well understood actually have a couple of mysterious characteristics embedded within them. And as an example, I draw your attention to this one histone modification catalyzed by the BUB1 protein kinase on a threonine 120 of histone H2A. I already told you that this modification is thought to emanate from the kinetochores and is one of those crosshairs that localizes the CPC. We can see this gradient of phosphorylation emanating from the kinetochores if we stain for this phosphorylation site in prometaphase cells. Here are those two marks coming out from the kinetochores. But now we need to capture that gradient of phosphorylation in a model that we're abstracting as follows. We have, this is a 2D model that we have set up in VCell. I'll show a little bit more details on that in a moment. It's not a compartmental model, but we have different domains where we specify certain reactions are permitted to occur or not occur. Species are allowed to exist or not exist. And the 3 domains of interest for this model is the chromosome, so the chromatin itself, this cohesin stripe that keeps the sister chromatids together. There are certain kinases that are restricted to this domain. And the one that's most relevant for this slide is the kinetochore, which is where certain scaffold proteins are that are involved in the recruitment of diffusible kinases such as BUB1. And the way that this is thought conceptually to work is that BUB1 binds a scaffold at the kinetochore, and that binding and release is sufficient to give rise to this gradient of phosphorylation because now it's closer to the substrate step. That was just the first 2 steps of this model, and these are some of the variables that we put in our V-cell encoding of it. What we quickly learned is that when we just did this binding and unbinding, because of those differences in diffusivity that I just showed in the slide before, simply recruiting BUB1 to the kinetochore and letting it go was insufficient to give rise to a gradient in the phosphorylation like was observed experimentally. And the reason is because of that very rapid cytoplasmic diffusivity that I touched on just a moment ago. It binds and releases, and then there's hardly any accumulation of phosphorylation in this kinetochore region in simulations. And so what this prompted was a revision to the model where we thought, or postulated that once BUB1 has been recruited, it likely has many options of phosphorylating histones because these are very abundant chromatin constituents once the kinase is close to the chromosome. And so what we premised was that BUB1, when close to the chromatin, would have more chromatin-like diffusivity. And the way that we modeled this was after the binding of BUB1 to this scaffold. When released, we added a new species to the model that we annotate as BUB1 his. And this is simply a less diffusive version of BUB1 that eventually can interconvert, be released, and go into the cytoplasmic BUB1 state. But BUB1 his has that histone, that chromatin-regulated, slower diffusivity that I mentioned previously. And when we do that, I'll orient you here on a couple of the simulation outputs of it. What did we start with? BUB1, cytoplasmic, diffusible species. It's everywhere in our model. I'll talk more. These void regions here are excluded from any part of the simulation. We localize that kinetochore protein right at the edges here, those landing pads for the mitotic spindle. And then in these simple simulations, we are just putting the H2A substrate in between the two kinetochores in the so-called inner centromere. So this is the way that the model starts, coated with this histone-bound species. What we find is that we get now a nice gradient of that histone-bound BUB1 emanating from the kinetochores and a corresponding gradient of phosphorylated histone emanating from the same site. But we only got here by failing in the earlier version that didn't have that histone-bound species. Now, that was a very early version of modeling this one step in the network and ignored any of the diffusivity of the chromatin or any of the broader regulation of BUB1 in mitotic cells. And one of the things that we have found in the literature is that BUB1 is constitutively active during mitosis, so it's active everywhere. And if we ran the full simulation, giving BUB1 access to all of the H2A that is all along that mitotic chromosome, what we found is that BUB1, when fully active at its cytoplasmic concentrations, is apt to phosphorylate everywhere, all along the chromosome, shown in these pseudo-colored images here. And when BUB1 puts this modification all along the chromosome, there's no longer an avidity effect right at the inner centromere, and you get this diffuse localization of the CPC that never bears any resemblance of the final recruitment that we see by imaging. This failure prompted us to go back in the literature, reminded us of another characteristic of histones in mitosis, and that is that they can also be acetylated and deacetylated. In fact, H2A is acetylated and deacetylated at a lysine 119, that is very close to the T120 that needs to get phosphorylated by BUB1. And what occurs is that most of the chromosomes, metaphase chromosomes, are acetylated, but there is a selective deacetylation of histone H2A right around that inner centromere kinetochore region. And this is important because the acetylation mark is so close to the threonine 120 mark that more likely than not, this acetylation is blocking the ability of BUB1 to recognize or phosphorylate this T120. And what this motivated then was a revision to the model where we restricted BUB1 activity only to that inner centromere region, like what I had shown before, but now incorporating the ability of, once phosphorylated the ability of that chromatin to diffuse within the simulation using those parameters that I touched on before. When we incorporate that change, we see a much nicer and more experimentally consistent gradient of phosphorylation of H2A. And then reassuringly, also a more teardrop intercentromere type of localization and recruitment of the CPC. Okay, another facet in terms of error model quality control checking, simple validations of the model. We wanted to evaluate what the relative importance were of the different loops and these different modifications that I specified earlier. Spoke a bit about that T120 modification, but there is another, this phosphothreonine 3 modification that occurs on histone 3, only along this cohesin stripe. And so we can run our existing model through various digital inhibitor or knockout type of experiments. We can set the catalytic activity of Haspin to 0. And when we do that, we no longer get intercentromere recruitment of the CPC. Either it doesn't recruit or maybe there's some minor recruitment to the kinetochores, but clearly disrupted as a result of removing Haspin. We observe a different phenotype if we fully inhibit the activity of BUB1. And this experiment can be done genetically in cells, knock out BUB1, I believe this is in a Xenopus chromosome. Localization of the CPC changes from the intercentromere to now this extended staining along that, that cohesin stripe. And I'll touch more on this droplet type of staining later on in my slides. And so those simple genetic-like experiments in the model were reassuring that we were ready to get to the, in many ways, the essence of what we were going after, which was how the CPC senses and becomes activated in chromosomes that are improperly attached, meaning that their kinetochores are close to one another, that 600 nanometer distance that I touched on before, compared to when they're properly attached and the kinetochores are under tension, and the separation between them doubles to 1200 nanometers. And what we asked, and what we're continuing to ask, is whether this separation of the kinetochores is sufficient to describe recruitment of the CPC in this state, and then the lack of recruitment or a de-recruitment of the CPC for a chromosome that's in this state. And what's been documented in the literature experimentally is that this separation coincides with differential recruitment. Misaligned chromosomes in normal retinal pigment epithelial cells or normal foreskin fibroblasts has about a 3-fold increased recruitment for misaligned chromosomes compared to aligned chromosomes. And what we're really interested about is in the setting of cancer, HeLa cells or U87 glioma cells, they lose the ability to differentiate misaligned and aligned chromosome by CPC recruitment. And I'll expand it a bit on why we think that might be and how we want to use the model to evaluate that. So we're going to test this now in our model. I've tried to tread lightly on the biochemistry that underlies these different domains, but here it is in a high schematic of different cascades of phosphorylation and scaffold-dependent recruitments. The CPC auto-activates, and we have that all encoded. And all of this is encoded in V-cell. This is, we don't show this hairball too much, but this is the schematic that we have that codifies those different species in this CPC model, either in the relaxed or in the tensed state. Take our V-cell model, run it through these two different scenarios, where the only difference is the increased spacing between the kinetochores here and the kinetochores here. And then we run it out on a time scale that's consistent with metaphase. We often use 500, 600 seconds or so. What we found was that early on, we see that in separated kinetochores, there is a mild difference in recruitment here compared to the bottom compared to the upper, but it's pretty minor. It's about 1.5-ish fold. And it gets even more muted as one goes later into the simulations. And what this result indicated to us is that we were missing certain facets of either the regulation, certain encodings of the reactions themselves, or maybe some other facets of the biology that weren't incorporated into the working model. And one of the ones that we first thought of is that this schematic that I showed in the preceding slide here was a bit artificial from the standpoint of what occurs normally during mitosis. I showed that video before as the nuclear envelope is breaking down and cells are in prophase and going into prometaphase. All of the chromosomes are improperly attached at some point. They all start at some state here. And what happens during metaphase is they all eventually transition over into this amphitelic state. And therefore, what we sought to do was capture this state switching in the model, going from a relaxed to a tensed configuration, and evaluate whether that might have an impact on the outputs that what the model was giving us. And our strategy was as follows. And this is where we started really pushing V-cell into new areas that we weren't aware of. And so I wanted to highlight this to give an illustration of the flexibility of V-cell and the types of things it can do beyond the simplest and very straightforward things that you might be getting introduced to during this workshop. And so what did we want to do? We wanted to run the simulation with improper attachments for a certain period of time, 100 seconds, 200 seconds. Take the outputs of that model and then feed them in as initial conditions to a new model that had that separated kinetochores and then run out from there. And what we wanted to evaluate was what happened at that state switching, depending upon whether we just kept them going with improper attachments, would they continue to increase their recruitment of CPC, and then critically, what would happen when there were the separated kinetochores. We envisioned a scenario where they could just stop recruiting, and then this would exaggerate the difference, like what I showed in the experiments before. Or maybe even we could start seeing a de-recruitment of CPC from the intercentromere. So these were the two mental models we were thinking about to motivate this change. To do this model input and output from a V-cell, we needed to use these outputs of V-cell called field data. And so the field data, you may have touched on this in your workshop already, captures all of the spatial species concentrations, time concentrations of the model as it's being simulated, and can be put in as starting conditions for a new model. And so this is what we've been doing during this transition. And for some of the species in the model, this was relatively straightforward. I'll give you a straightforward example, which is one of the kinetochore scaffolds called NDC80. Here's where it's localized in the relaxed configuration. It's restricted to the kinetochore, doesn't move anywhere, and doesn't change very much during the simulations. And so if we want to update where it should be when the chromosome is properly attached, we simply just relocate it out to that stretched configuration. That's a simple example. We had many more that were complicated because they were already 100, 200 seconds into the simulation and in different bound states within that inner centromere. Notably, all of those CPC bound states that I've been showing schematically up to this point. And so it was about this time I grabbed this slide for a reason, because it was one of the slides that I presented at UConn during that visit that Jan talked about it in the beginning during my introduction. And so we were struggling with ideas about how we could relocalize these chromatin-bound species when now all of a sudden there was double the real estate in between the kinetochores. And in fact, the chromatin reduces its concentration by half just by mass balance, because now there's spread out over a larger volume. And so we were thinking about all these different ways where we could have this and move that around and kind of heuristics and things. And I'll thank Les here for raising his hand and telling us to stop doing any of this and look into the capabilities of V-cell to encode a process called advection to literally move species with a velocity specified in V-cell to different locations in the simulation. which is what we did. And learned some things along the way. So my title is called Making Mistakes with Advection. So this is a fun way not to do it, which I wanted to share. And so we looked into to the advection capabilities of the V-cell. This is a reminder of where we started. So run the simulation here now for 100 seconds, export the field data, move it into the newly localized kinetochores. And then we said, okay, we have to deal with all of these CPC-bound species in the inner centromere. Let's use advection. This is a bulk motion effect. Here's one version of the equation where you have a velocity and you have your for a concentration, and then you advect, you move that concentration depending upon the velocity field that one specifies in X, Y, or Z. I said, oh, this is really easy. We don't even have Y and Z of interest here. We simply want to move everything in the inner centromere along X to the left, and on the right-hand side to the right-hand side. Specify a velocity. We think it happens really fast, so just move it. And this is what happened when we did that. When we specified a constant velocity field in the advection term for all of those species, we parted the waters and just split the CPC bound states without anything in the middle. We can quantify this with a chymogram. We do a line scan across here, and the essence of what we were erroneously doing is we were moving the species too fast relative to that chromatin diffusivity that I talked about before. And the model doesn't know any different. It'll just move it and spread it out. But we were creating this huge void in the middle as a result of moving way too fast the species and way too uniformly over the time scale that we were doing it. We had to find a different way. And how did we do that? Well, we admitted we had to make a more complicated velocity field. I apologize for the color encoding here. This is just what V-cell can pseudo color. So 0 is green. So everything here, there's no velocity. And then what we did is then defined a velocity that was dependent upon where a species was located along this X coordinate. So the exact midpoint, nothing moves. And as a species moves further and further out horizontally, it moves faster up to the maximum, which is set as just the velocity needed to move the outermost species to the end of the stretched kinetochore, as indicated here. For us, this ended up working out really well. Here's a little GIF here of each one of those frames as we advect the species out with this distance-dependent velocity field. And here's the same chymogram that I had shown before with a much smoother stretching of the CPC-bound species over the timescale that we did it along. We can take individual line scans here and see by and large Gaussian type of distributions. There's maybe a couple of shoulders here, but overall we were pretty satisfied with this ability to stretch out everything in the inner centromere in a principled way that didn't create or destroy things or involve too many heuristics about what we think, how we think that stretching might work. Okay, so then this is how we updated our model, exactly as I had said, piecing these different things. And now let's run the simulation after the inner centromere has been advected laterally. The first thing that we noted, so this simulation now starts at 100 seconds because it's been preloaded with 100 seconds of simulation from before. First thing that we saw is that stretching of the chromatin gives rise to a really dramatic reduction of CPC localization per unit area because we've just halved everything by stretching it out. Second, we noticed that even after that simulation moved ahead, there is a recovery phase where the CPC continues to accumulate even in the stretched case. We can show this quantitatively. Let's focus here just on the active CPC state. So it's trajecting upwards here to a very high concentration. We stretch it down, and then it moves into this stretch state that is lower, but it's still continuing to accumulate. And if I circle back to what I originally had said as the two scenarios, it was really neither of these. In fact, what we appear to be seeing is a dip and then a rebound. And this rebound is something that we're continuing to think about whether we fully captured in our model. Just in the final, how am I doing? 5 minutes or so, I wanted to talk about some things that are in progress related to this and in development stages, including in partnership with some of the B-cell folks. Just transitioning from the last thing that I showed before about this CPC rebound, one of the things that we're considering is that one could still get a very dramatic change in effective CPC concentration if there were some type of biophysical event that was a thresholded event where if the recruitment was below that threshold, something wouldn't happen, and then if above that threshold, something would. And what we believe that something might be is the formation of a bimolecular condensate involving the CPC. So Todd's group a couple of years ago now had shown that when the CPC is recruited to a critical concentration under physiologically ionic strength conditions, the CPC can form a condensate, phase-separated type of liquid droplet, a membraneless organelle. And the thinking is that this occurs right at the intercentromere because of those crosshairs that I talked about before, and that this phase separation provides some degree of buffering, capacitance, if you will, to form even if there are fluctuations in some of the species, until it doesn't form, and then there's a really catastrophic event like a chromosome missegregation. And so what we've been doing, and this is in collaboration with John Lone Group at UC Irvine, he's actually my 1 o'clock meeting, so we'll be sure that this finishes to time, is using some numerics from the chemical physics literature several decades ago by these fellows, Kahn and Hilliard, about modeling fluid phases and energy minimization of phase-separated systems. I'm not saying anything too much more than that, but I'll flash you an abbreviated version of the equation that describes a chemical state, a droplet state or a liquid state in this application. And what I want to emphasize is that numerically, this type of formalism is challenging to solve computationally because this Laplacian of a Laplacian here creates a fourth order spatial derivative that is very unstable. So John is really an expert in numerical methods to be able to arrive at numerically stable solutions to this type of equation. And I'll show one video if we take this as a simulation of recruited CPC and then everything else being in the bulk, and we say undergo a phase separating type of process. What happens? We see these droplets starting to form, and then these smaller droplets undergo a process called Ostwald ripening, which occurs when the droplets are too small to maintain the interfacial energy between phases, and then they just lose mass into the bulk. And this was really striking to us because let's take that same movie and just splay it out frame by frame. A very similar process happens with the CPC. Early in prophase and early even in some of our own results, one can see these types of droplets forming along that midline. Remember, I talked about that when I was highlighting a Xenopus chromosome before. And then as those cells progress into prometaphase and metaphase, those droplets disappear. And so we're really excited about incorporating a new compartment that's informed by this phase field modeling related to the bimolecular condensate. We think that this may provide that switch-like character that I showed schematically in the earlier slide. And then finally, I wanted to circle back and finish with the tie of this to cancer. Everything that I've shown up to this point has been nominally normal CPC regulation and trying to capture it. But where we want to go is look at how this process gets misregulated in breast cancer specifically, and a subtype of breast cancer that's highly aneuploid. Todd's group has worked on this several years ago, looking at the genetic characteristics of aneuploid breast tumors. And what I want to highlight are some of the differential expression patterns that were associated with highly aneuploid tumors that had undergone chromosome missegregations at some point in their lifetime. The big surprise was the large number of mitotic regulators whose abundances were very different in aneuploid cancers rather than ones that had lower levels of aneuploidy. In cancer, none of the constituents of the CPC network are mutated. And the thinking is that they can't be, because if they're always on or always off, then the cell wouldn't proceed through mitosis and you'd have no cancer. Instead, what might be happening is that the initial conditions are set up differently different abundances of kinases, scaffolds, and so forth, so that then the network becomes error prone. Maybe it doesn't sense those tensed and relaxed transitions like I had shown from before. And so what we would ultimately like to do, what we're asking is, can we use our V-cell model as we continue to refine it to predict the impact of these types of abundance changes just purely by incorporating different initial conditions? Let's say for all cancers, breast cancers in the Cancer Genome Atlas. And then the Cancer Genome Atlas is about 1,000 of those. Now, 1,000 simulations in the graphical user interface of V-cell is prohibitive. Nobody wants to do that. And so this has been a motivation for us from pivoting from the V-cell GUI that I'm sure your workshop is based on to the command line interface, some of the work that Jan alluded to before. The graphical user interface is great because you can build a model and it's very accessible. But as one starts moving ahead and really kind of wrangling the data, we had to do some changes with how we handled exports from the simulations. So we've done some things with HDF files to be able to wrangle them a little bit easier, get them in a ways that we could show graphs in whatever way that we wanted. And what it pointed to us is we really needed moving out of the graphical user interface and into the command line interface, which is possible. We have a supercomputing cluster here called Rolana that we've been able to run simulations for, and then they can done in parallel. And so we can allocate a certain number of cores per simulation and then run 500, 100 different simulations out to various cores and then collect the data back at the end. Now, a little bit of a drawback, Jan said this, we're the beta testers of it. So we are working with the V-cell team to identify bugs or places where it works, or the simulation runs and we can't get the output. But it's something that we're actively in dialogues with them. And then the last little bit that I'll just mention is that one aspect of running V-cell on the command line interface and doing something like a very large parameter scan, 1,000 different initial conditions or so forth, will eventually require us getting under the hood of V-cell and manipulating this file called an omics file. Last slide, just to show the difference between the V-cell graphical user interface and what an omics file looks like. This is a zipped file that has a manifest in it. And the key business end that Jan and his team graciously taught us about is this markup file called a setML file, which contains all of the relevant and required information to run a simulation. So which ones have been run, what kind of models, task generators, et cetera. This is a little snapshot of what it looks like. And diving into that setML file enables one to then manipulate it and change certain parameters and task jobs accordingly. And so this has been our strategy to be able to run a large number of V-cell simulations in a distributed environment. I'll stop there, and I'm going to thank the two modelers on our team. Catalina has spearheaded a lot of the original model construction, and Sarah has been instrumental in helping with that tense to relaxed transition, and she's also actively working with John and a postdoc of his at UC Irvine on some of the phase separating behavior. And then finally, I want to acknowledge the early undergrads. This team came together before we had a center. We had just Todd and me and a pair of undergrads, Lydia and Jazraj, that really got the first versions of these V-cell models up and running. And then Mia came in for a summer to help maintain momentum. And so I just draw attention to that because it really speaks to the accessibility of V-cell as a tool that can start very simple, start with accessibility, but then rev up in its numerics as you go ahead. And I'll thank the V-cell team. Jan, Logan's been very helpful with some of the command line interface. Jim worked with us when we were there in person in the fall. And then Les, I won't forget the advection recommendation. And I'll stop there. Thank you.
Thank you, Kevin. This was great. You know, pretty much using all the tricks of the trade to do this.
Any questions?
No, I just wanted to point one thing out that while I like the advection better for the way you handled it, in fact, the most recent code that was left to us by Frank Morgan does allow for scaling and translation of the field data to fit a new geometry. So if you find you need that in the future, just give us a buzz and I'll dredge that up. That's in the alpha version. It's the only place it is. And I haven't actually played with it that much, but it's there.
Interesting. So it's called scaling and translation.
Yeah, and
Okay.
at this level, it was the last thing that Frank did before he retired. So it's like a hand-drawn T is the little symbol for the tool. So it's very crude, but it does actually will scale and translate the field data to fit a different geometry than the field data was originally done with.
Oh, interesting. Good to know. Because I think we also have there may be variations of this that may be harder to capture with an advective field.
Right.
And yeah, so we may be circling back on that because there's a couple
Okay.
places where it's great for now, but I'm wondering the final version might need to be more flexible than that. So that's terrific. Thank you.
Yeah, just
Yeah.
related to that, if you have another second, in those simulations where you use the advection, it looked like the CPC above and below obviously hadn't moved because they hadn't been advected. So is that a problem? I mean, should they have also been subject to some sort of mild advection?
This is precisely the limitation that I was alluding to in my response to Anne. And so, well, if we do, well, how high do we go? And so for just simplicity, we kept it in between the two kinetochores. But if there's a scaling and translation where we could specify some more elaborate and probably more authentic geometry there, we would be very interested to explore that. I don't think it will change the salient characteristics of what we have in the network at the moment. But in a final version, if we had a cleaner way to specify that, then that would be really desirable.
Yeah.
You
And-
haven't
Well, you
published
could
any
use
of
a
that,
Y
Kevin.
dependency in your function.
Yeah,
For
you could
example.
just do that. Yeah, you could
Yep.
do.
Mhm.
You didn't publish any of that yet, did you?
No, my meeting just before this we're right at the start with Catalina and Sarah. We just had our first brainstorming session about what figures should start to look at. And so it's timely that this presentation, we're hoping to be working very hard on this in the spring into the summer. I'd hope we'd get something by the end of the summer.
I also have a quick question. I noticed in one of your slides that at the right end, it seemed to be some reflecting effect, a little tip up in concentration. Is that the boundary effect or?
Yes, only you, Jan, would see that little blip. We noticed it also. I take it as a boundary effect, and we kind of slam it into the wall. And if there may be a rounding error or some other types of things where it just accumulates there, and you see it reflected in that line scan. Again, another aspect of I think it's workable now, and that likely smooths out in short order once we move ahead from there. But if something like this scaling and translation is a more flexible way to do it, or there's other tweaks for refinement, we're definitely open to considering them.
Yeah, it might also be some weird artifact of the distance to center expression, would need a little bit more thinking, obviously. Would love to dig into that.
It could, for example, be just a grid point. It could just be off by one grid point, the way that Sarah rounded it out from there. I don't think it's a numerical art, some sort of like numerical
probably
artifact.
Not. It may be a voxel size issue or
Yeah, you should definitely use a finer mesh as just a sanity check to make sure that it isn't simply numerical.
Yep. Yeah, we've avoided doing that for other reasons, but to simply increase it on a one-off run and evaluate whether it makes a difference is a good recommendation, certainly.